also install values, confidence, and a love for learning. Your patience, your mentor, a motivator, and a role model. Your commitment to shaping the future through education is truly commendable. And this award is a well-deserved honor. Congratulations on receiving the Best Teacher Award to Dr. Ramesh Singh Sir, Direct BLCP Lucknow. Thank you, sir, for your tireless efforts, your belief in your students, and for making a difference in their lives. Your impact will be felt for generations to come. Last but not least award is Pharmaceutical Award. It goes to Professor Dr. Ashutosh Mishra sir, Principal A&D College of Pharmacy, Patna. Please give a big hand for sir. Heartfelt congratulations, sir. Your dedication and contribution to the pharmaceutical industry have rightfully earned you the prestigious Pharmaceutical Award. It is a well-deserved honor. Your efforts in advancing education, research, healthcare, and innovation have made a lasting impact. This recognition is a true testament to your excellence. You are inspiring excellence. Your passion for innovation and patience with being has set a remarkable benchmark. This award reflects your incredible journey of dedication and perseverance. Thank you, sir, for inspiring us. I would like to request Professor Dr. Dhiren Kumar Gautam sir and our chairman sir Dr. Ranjan Mishra sir to felicitate our special guest Karuna Shankar Sukla sir. Sir, please come on the stage. I would like to request Mr. Shubham Tiwari sir and Professor Dr. Giren Kumar Gautam sir to felicitate Professor Dr. Amit Nagar sir. Sir, please come on the stage. Please give a big hand together. We are privileged to have another aristocratic personality with us today, Dr. Ranjan Mishra sir, Chairperson of TRC Mahavidyalaya Department of Pharmacy, who has played a crucial role in translating vision into action. So please come on the dais.
थैंक यू डियर एंकर्स, आप सभी को जय श्री राम। यहाँ पर उपस्थित जितने भी लोग हैं उसमें से मैं अपना वंदन उन शिक्षकों के प्रति करना चाहता हूँ जिनकी वजह से स्टूडेंट्स भी उपलब्ध हैं और ऑब्वियसली अब वो शिक्षक जो बड़े पदों पर आ गए हैं लेकिन उनका मूल है ही शिक्षक। तो उन सभी के लिए गुरु ब्रह्मा गुरु विष्णु गुरु देवो महेश्वर गुरु साक्षात परब्रह्म तस्मै श्री गुरुवे नमः। तस्मै श्री गुरुवे नमः। I am really very proud to have this type of platform in my campus. Rather, using my word would be very much mean. Your presence has made me to say that this is your campus only. इससे पहले मैं आकाश जी से कई बार मिला हूँ, कई ऐसे विषयों पर चर्चा हुई, लेकिन इस कॉन्फ्रेंस को लेकर नहीं चर्चा हो पाई। उन्होंने बहुत शॉर्ट टाइम में कहा कि हाँ मेरी प्रेजेंस तो 100%, आप ये समझिए मैं ही होस्ट रहूँगा। तो अपने कीमती समय से यहाँ पर समय निकालना और समय देना, बच्चों को आशीर्वाद देना, आप सबसे इंटरेक्ट करना, इसके लिए मैं आकाश को बहुत बहुत धन्यवाद देता हूँ। इस कॉलेज के बारे में आपको बताता हूँ। ये जो कॉलेज आप देख रहे हैं ये वास्तव में एक यात्रा है। ये यात्रा एक शिक्षक की यात्रा है। मेरे पिताजी शिक्षक रहे, यहाँ मैथमेटिक्स के शिक्षक रहे, और मुझे लगता है कि मेहनत का कोई और पर्याय नहीं होता। उन्होंने अथाह मेहनत करी, अथाह, और मैथ्स के साथ उनकी ट्यूनिंग इतनी थी कि if I say him that he was the synonym of mathematics, तो गलत नहीं। He used to count numbers like anything। उनको बुक्स याद हो गई। जब वो पढ़ाते थे तो वो कहते थे कि फलानी बुक में देख लेना, इस वाले प्रश्नावली 5 में चौथा प्रश्न जो है वो फलानी बुक में प्रश्नावली 10 का 8वां प्रश्न है। और जो स्टैंडर्ड उस समय IIT और अभिनार का था, he used to count them on fingers। मुझे याद है कि मेरे भाई साहब वो इंडियन फॉरेस्ट सर्विसेज की तैयारी कर रहे थे, उनको कोई प्रश्न नहीं आया, तो उन्होंने पिताजी से कहा कि ये वाला क्वेश्चन नहीं आ रहा। क्वेश्चंस वेर टिपिकल नो डाउट, तो उन्होंने कहा टाइम को थोड़ा सा मैं चेक करता हूँ। उनको वो क्वेश्चन 2-3 बार सॉल्व करने पर भी नहीं है, मेरे पिताजी को भी। he was so much eager, उन्होंने कहा चलो खाना पीना खा रहे हैं और वो सो गए। सवेरे जब मेरे भाई साहब उठे तो उन्होंने कहा कि वो सॉल्यूशन मिल गया उसका। तो भाई साहब ने कहा कैसे मिला है? तो उन्होंने कहा मैं सो रहा था तो मुझे अचानक लगा कि जो फॉर्मूला मैं लगा रहा हूँ वो सही नहीं लगा। See, he was thinking the same question in his sleep also. तो शायद वो डेडिकेशन टीचिंग प्रोफेशन का और स्टूडेंट में ग्रैबिंग का कुछ मिस हो गया, कुछ। अभी जैसे सतीश सर ने कहा कि मेहनत की बात हुई और एआई की बात हुई, बहुत सारी चीजें हुई। मैं आप लोगों का बहुत ज्यादा समय नहीं लूंगा, ऑल्दो आई हैव रिटन मेनी पेजेस, स्टिल मैं कोशिश करूंगा कि मैं जल्दी समाप्त कर दूं। जैसे सतीश सर बता रहे थे कि उन्होंने NSR 3 चीजें बताई, दैट इज नेशनल एजुकेशनल पॉलिसी, और फिर सस्टेनेबल डेवलपमेंट और रेवोल्यूशन की बात करी, ये इंडस्ट्रियल रेवोल्यूशन की बात हो रही है। उन्होंने आकाश सर ने बताया जैसे कि AI भगवान, सर ने भी कुछ मिलती हुई वैसी ही बात करी और उन्होंने बताया कि प्रैग्मेटिक नॉलेज, कल्चरल नॉलेज, इसको मिस नहीं कर देना। AI को वी शुड, जैसे हम लोग का पुराना स्टेटमेंट है कि साइंस एज अ गुड मास्टर और अ गुड सर्वेंट, तो वी हैव टू यूज साइंस एज अ गुड सर्वेंट, नॉट एज अ मास्टर। हाँ, भगवान अगर बना लेंगे तो ऑब्वियसली यू विल बी वर्किंग एज स्लेव। जो नंबर ऑफ रील्स चलती हैं अनडाउटेडली हम लोगों को भी नहीं याद होता कि कितनी रीलें देख रहे। फिर आशुतोष सर, नो डाउट, प्लीज एनवे मत फील करिएगा, ही इज वन ऑफ माय फेवरेट्स। मैं उनसे 2-3 बार ही मिला हूँ, लेकिन हमारी उनकी बात शायद आंतरिक बहुत ज्यादा होती है। मैं भगवान शिव का भक्त भी हूँ और उनका भी नाम आशुतोष ही है, तो कुछ अटैचमेंट उस वजह से भी हो जाता है। तो जो भी टाइमिंग की बात हो रही है, जैसे हम लोग घड़ी बांधते हैं लेकिन वो उस घड़ी की एक भी घड़ी हमारे कंट्रोल में नहीं है, तो वो उस चीज को मैं पॉइंट आउट करते हुए आई वुड रिक्वेस्ट दैट वी शुड स्टार्ट अ टेक्निकल सेशन। एंड थैंक यू ऑल, थैंक यू वेरी मच फॉर योर टाइम एंड प्लेजर, थैंक यू वेरी मच।
Let's begin with our first technical session. For that, we would like to invite our eminent speaker, Dr. Dinesh Kumar Sir, Additional Professor, Department of Advanced Spectroscopy and Imaging Center, Biomedical Research, Lucknow, Uttar Pradesh. And our chairperson, Dr. Divyan Singh Sir, our chairperson, Dr. Deepak Pandey Sir, for the first session. I request to all the dignitaries to kindly occupy their honorable position at down the stage.
invite Mr. Mohammad Ubaid sir to give an introduction of Dr. Dinesh Kumar sir.
तो दूसरा प्रोग्राम हो जाएगा अभी
पोस्टर
कौन सा
पोस्टर प्रेजेंटेशन
मतलब
इसमें अभी स्लाइड करेंगे तो अगर लोग वहाँ पे बताएंगे
ये इसमें ये पीछे तो चलो चलो फोटो भी लेते हैं पीछे तक लगे हो
पूरा ले लिया
चलो एक बार
चलो
चलो आओ
इधर लाइव चला रहे हैं
अच्छा अच्छा
I would like to request Professor Dr. Virendra Kumar Gautam sir and Mr. Shivam Tiwari sir to felicitate Dr. Viren Singh sir. So please come on the stage. Please give a big hand together.
वहाँ तो यहाँ प्रोग्राम चालू हो जाता है स्टैंड अच्छा लगता है
आज से कितने इधर
डायरेक्ट मोबाइल तुम स्कूल के स्टूडेंट डायरेक्ट मोबाइल के तो बनाओ डायरेक्ट फ़ोन के तो बना लो
अरे यार
प्रिंसिपल कौन है
Let's begin with our first technical session. For that, we would like to invite our eminent speaker, Dr. Dinesh Kumar sir, Additional Professor, Department of Advanced Spectroscopy and Imaging Center, Biomedical Research, Lucknow, Uttar Pradesh. And Dr. Viren Singh sir, our chairperson, Dr. Deepak Pandya sir, for the first technical session. We would like to invite Mr. Mohammad Ubaid sir to give an introduction of Dr. Dinesh Kumar sir.
Good afternoon, all of you. Myself, Mohammad Ubaid, Associate Professor, PRC Mahavidyalaya, Department of Pharmacy. Today, jointly organized International Conference, PRC Mahavidyalaya, Department of Pharmacy and Peshawar. Introduction of Dr. Dinesh Kumar sir. Dr. Dinesh Kumar sir is an additional professor in the Department of Advanced Spectroscopy and Imaging at the Center of Biomedical Research in Lucknow, Uttar Pradesh. With his extensive expertise in the field of biomedical research, Dr. Kumar sir specialized in advanced spectroscopic technique such as nuclear magnetic resonance, spectroscopy and its application in clinical metabolomics. Dr. Kumar sir earned his PhD in chemical sciences with a specialization in solution state NMR from the Data Institute of Fundamental Research in Mumbai. He has been associated with CBMR since 2010, serving as a research associate, assistant professor, and later as an associate professor. He has been a part of CBMR since its early years, beginning as a research associate and later progressing through roles as assistant professor and additional professor. His progression highlights his dedication to advancing scientific knowledge in the biomedical fields. Dr. Kumar sir research in focused on applying cutting-edge spectroscopy techniques to study metabolic profiles in biomedical systems. Specifically, he used high-resolution NMR spectroscopy to investigate metabolic alternators in various pathological conditions by analyzing the metabolic signatures of disease. His first time to identify biomarkers for early diagnosis and therapeutic intervention. Dr. Kumar sir works in clinical metabolomics, play a vital role in developing non-invasive diagnostic tools. His research in contributing to a better understanding of disease mechanism and the discovery of novel therapeutic targets. His work has the potential to significantly influence the development of personalized medicine, where treatments and tolerate based on individual metabolic profile. Dr. Kumar sir is also recognized as a fellow of the Indian Academy of Science, which is acknowledgement of contribution to the scientific community. Additionally, he works as position him as the leader of application spectroscopy in the study of complex disease. He has received multiple grants and awards further supporting his research efforts. Thank you.
Now it's time for second technical session sound. Thanks to you, Mr. Augusta. I express my gratitude to you. Thank you, sir.
Thank you. I would like to say my heartfelt thanks to all the members of the organization, especially Dr. Kumar sir, for inviting me to this event and giving me the opportunity to present some of our research in this event. So, let me start with a brief introduction. So, currently I am working at Center of Biomedical Research.
Lucknow. That our campus is within the SDPJ campus, Lucknow. So, at CIMA we have three high-field analog spectrometers, 400 megahertz, 600 megahertz, and 800 megahertz. All three spectrometers are having their own capabilities and used for different purposes. I really work on 800 megahertz high-field analog spectroscopy. So that we use for structure studies of proteins, and the information is further used for rational drug design. And further, we are using this high field analog spectroscopy for performing the clinical metabolomics analysis. So, actually here, I'm not sure that whether all the participants here are aware of analog spectroscopy, but that itself is two-class lecture to make you understand about analog spectroscopy. But in simple words, you imagine that this is spectroscopy technique which helps to quantify or give information about the structural or dynamic features about some molecular structures, molecular compounds. Okay. And over the years, the analog spectroscopy technique has flourished dramatically over methodological and technological fronts. MR today has become one of the most versatile tools for almost in all the aspects of biomedical and pharmaceutical research. Whether you are involved in evaluating therapeutic efficacy, safety of potential candidates or the formulations, you may require animal spectroscopy at one or other steps of that whole procedure. And if you are involved in some clinical research, then also you may require animal spectroscopy or clinical MRI at one or other stages. And also in pharmaceutical research, it is indispensably used by pharmaceutical companies, this animal spectroscopy. So, So now let's first try to learn what is metabolomics. Okay. So let me make the lecture very simple for all you students. So we have various omics techniques in our research setup, like genomics, proteomics, transcriptomics, now epigenomics is there, and metabolomics. Mainly in our lab we do metabolomics. Okay. First, let me tell you about what is proteomics. Quantitative and comparative analysis of proteins expressed in a biological system is known as proteomics. Okay. Now, just throw the proteins out and put metabolites. Quantitative and comparative analysis of metabolites present in a biological system is known as metabolomics. This is clear to everyone? This is clear. Quantitative and comparative analysis of metabolites present in a biological system. Okay. Suppose in my body I have glucose and glucose is elevated. I have hyperglycemia. So that is a metabolite. If glucose is elevated, I may have a clinical condition like diabetes or some other clinical condition. Okay. So in our research setup, we try to see in a particular disease condition what are the metabolites which are elevated, what are the metabolites which are decreased, and based on those metabolic patterns, we try to identify the severity or type of clinical condition. So this is what the main idea of performing the metabolomics. Okay. Now we have in our lab we do both clinical and pre-clinical metabolomics. Pre-clinical means when we perform this metabolomics analysis on pre-clinical models. Pre-clinical model is either rats or rabbits or some other animal models where they were induced with some disease, and we try to see a effect of the drug formulation or some potential candidate drug. Okay, so that comes under the category of preclinical metabolomics. In clinical metabolomics, we try to identify disease-specific signatures, and then we try to see if those signatures can help us to monitor the disease, whether disease is progressing towards its severity, or whether we can check whether the patient is responding to some kind of treatment, that is treatment monitoring, or to assess the severity of the disease, that is prognosis. And diagnosis is one of the main aspects of that clinical metabolomics, that is to identify disease-specific metabolic signatures. Means what are the specific metabolic patterns associated with a particular kind of disease. Next. I'm audible. Audible? I'm audible. Okay. So now in our lab setup, we use this metabolomics approach for disease metabotyping. Disease metabotyping means to identify disease-specific metabolic signatures and then try to see if those can be used for disease management. Okay, so in our setup, we are involved in disease metabotyping of various complex human diseases, including a range of autoimmune inflammatory diseases. So, an autoimmune inflammatory disease means when our human body starts targeting its own system, its own cells, particular type of cells, particular type of organ tissue. When our own body or our immune system starts targeting on human cells, okay, own cells. That is comes under the category autoimmune diseases. Next. So globally there are 108 types of autoimmune diseases. Next. Yeah. So there are more than 100 types of autoimmune diseases, and the common feature of next career. The common feature of all these autoimmune diseases is that the immune processes periodically becomes active, and after some time these processes become dormant or inactive. Okay, and these cycles of active inflammation and remission, or you can say inactivation, these keeps on going throughout the life of the patient having autoimmune condition. And as these cycles keep on going during the life of the patient, the disease keep on progressing towards its severity. And if the active phase of the disease is not managed appropriately, the disease progresses very rapidly. So, in the management, in the clinical disease management of various autoimmune conditions, is that to identify when the disease is active and to manage that active condition. So there is no cure for autoimmune disease because we cannot kill our own immune system to cure some autoimmune condition. So mostly these patients, when we found that the patient is having active disease, so those are given immunosuppressants so that immune system gets suppressed for some time and the patient does not feel the condition does not progress towards this well. However, getting this information, whether the disease is active or inactive, that itself is a challenge in clinical settings because during the initial phases of the disease, this activity can be assessed with reliability with some chronic inflammatory markers. However, after 1 or 2 years, these markers doesn't work and it becomes very difficult for clinicians to assess the disease activity when the disease is active. Next. So there are clinical markers like erythrocyte sedimentation rate, ESR and CRP. Those are used initially for assessment of disease activity. However, after some time they fail to assess the disease activity. Therefore, in clinical settings, clinicians require some objective biomarkers which can provide the reliable information about the disease activity so that the patient can be served with appropriate treatment. Next. So, for various autoimmune disease conditions, there is a particular kind of scoring system, empirical scores, based on the clinical symptoms, patient history, and what are the other clinical investigations with information. Based on those information, the clinicians calculate a score. Based on that score, they come to the conclusion that whether the disease is active or it is inactive. Next. So it is a very complicated process. Getting the clinical score, it may take 2 to 3 hours for clinicians to evaluate those scores based on the patient clinical investigation for clinical conditions. Next. 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 So now, this is one aspect when we are working on autoimmune disease. We need some objective biomarkers to assess the disease activity. Further, we need some metabolic signatures which can help us to monitor whether the patient is responding to that kind of therapy or treatment, and whether we can predict the relapse or a patient is really on the remission to that treatment, monitor treatment response, and sometimes autoimmune disease conditions also involve other organs. So comorbidity is also present, and also we need some kind of signatures to identify whether the patient also involves some other organ. So there are various aspects which are required in clinical settings when we have to manage the autoimmune disease conditions. So this is just one example. Takayasu arthritis. Takayasu arthritis is a rare autoimmune disease condition which involves granulomatous inflammation in the aorta and its vein branches. So that happens because some unknown reason. Therefore, the etiology is not clear why that happens with some of the patients. It is rare and mostly it affects the female population in Asian continent. So when that condition progresses, next. So in this autoimmune disease condition, also that happens with inflammation becomes active, and then it goes on, it becomes active, goes on, and as these cycles of inflammation and healing goes on, the aorta, the wall of the aorta becomes thick, and this lumen, where through which the blood has to go, that becomes narrow. So when this disease progresses towards this severity, the blood supply to the target organ gets occluded, and the patient may suffer from organ damage or organ failure if that organ is carrying blood towards that organ. So in order to to save the life of the patient or save that failure of that organ, the only method is to place the stent after surgical procedure. So when surgeon has to perform the clinical procedure to place the stent, so disease should be in its inactive phase. Disease should not be active so that the surgical outcomes can be favorable or in favor of the patient. Next. So this is the main reason why when the surgery has to be performed and stent has to be placed where there is occluded blood supply. So this assessment of disease activities becomes very crucial. Next. So other aspect is patients cannot be given immunosuppressive therapy because giving that immunosuppressive therapy is not good until unless you really have the disease in its active phase. Next. So next. Therefore, we need some kind of metabolic or some kind of biomarkers which can help us to assess the active phase of the disease. Next. So here we have used NMR based metabolomics, means metabolic profiling of blood serum, and we compared the blood metabolic profiles of active and inactive tachycardia arteritis patients. And next here. And we found that the metabolic profiles of active patients are significantly different from the inactive patients. And there are many metabolic profiles which are changed during active disease condition. And among all, the N-acetyl glycoprotein signal found to be significantly predicting the active phase of the disease. And we found that N-acetyl glycoprotein signal that also correlates with the clinical score, that is ITA-ASR. ITA is the Indian Arteritis score, which is calculated to some clinical investigations and some empirical formulations in empirical formula. And it is further has been improved by ESR score. So the clinical correlation of this N-acetyl glycoprotein signal that suggests that there is some clinical relevance of this signal, NMR signal. So next. So further evaluated the diagnostic. This is the receiver operating characteristic curve analysis. That is you may say advanced statistical method where you can assess clinical parameter or biological parameter whether that can predict the disease condition or that can predict some kind of clinical condition. So we found that the area under the receiver operating characteristic are also performed very well. That means this N-acetyl glycoprotein signal has the ability to predict the active disease in case of arteritis patients. Next. So in follow-up studies also it has been shown that this NMR signal has the ability to predict the inflammatory condition in other kind of disease, autoimmune disease and chronic inflammatory diseases. Next. So this is the signal which I have been talking about. The signal in the serum NMR spectra 2.01 ppm, and this signal represents the cumulative NMR signal of N-acetyl glycoproteins, and this signal has the ability to predict whether the patient is having active disease or it is inactive. Next. So other aspect of when our immune system gets activated, though the floating immune cells they start using glutamine at higher rate, and they are less dependent on glucose. Therefore, the glucose accumulation or slightly hyperglycemic condition builds in the circulatory system. And based on that, we hypothesize that if that is the metabolic paradigm or metabolic signature of autoimmune diseases, so glutamine to glucose ratio should predict the disease activity. So next. So we checked this hypothesis and we found that indeed the glutamine to glucose ratio is decreased in case of active disease patients, and also this has the ability to predict active disease in this group of patients. Next area. Next. So also next area. And also we have evaluated the circulating glutamine levels in other autoimmune diseases, and indeed we found that the glutamine levels significantly decrease when the disease becomes active. Next. And also this also provides a quantitative information. As the disease is highly inactive, the glutamine levels are significantly decreased. Next. Next. So this is another example where we have used NMR-based metabolomics for differentiating sarcoidosis from tuberculosis. So next area. Sarcoidosis is another autoimmune disease condition. Next area. Next. So that clinically or the clinical and radiographic features of this sarcoidosis, they are very much similar to tuberculosis patients. So in clinical settings, it is always a challenge to differentiate between sarcoidosis and tuberculosis. And next area, and in our country there is very high burden of tuberculosis. Most of these sarcoidosis patients, they are misdiagnosed and get the treatment like TB patients, anti-tuberculosis treatment. Next area. Next. So almost 30% of the sarcoidosis patients, they are misdiagnosed and they receive the anti-tuberculosis. But the difference between this tuberculosis and sarcoidosis is that sarcoidosis is an autoimmune condition and TB is an infection. When these sarcoidosis patients, they are treated with anti-tuberculosis treatment, the disease does not manage and start progressing towards severity, and sometimes lungs may get the disease may progress towards fibrosis. So that is why it becomes very important. Those patients are diagnosed appropriately or perfectly so that they can get proper treatment. Next. Next one. So, in order to perform the suggestive diagnosis of sarcoidosis, only procedure is to rule out the tuberculosis. To rule out the tuberculosis is that one has to perform the image-guided lung biopsy to get the samples from 3 to 4 locations, random sampling, and perform the next study. Next. So once you perform this biopsy, lung biopsy, it is very painful procedure. No sarcoidosis patient want to undergo this kind of procedure. So, but once one has to perform this procedure, the samples are taken from 3 to 5 granulomatous locations, and those are sent for microbiology testing and microscopy, where they rule out whether the granulomas are caseous or non-caseous, because the tuberculous granulomas are caseous. They have cheese-like structures inside, and whether those granulomas are sterile. Once non-caseous sterile granulomas are confirmed, that means the patient is most probably having sarcoidosis. So this is how the diagnosis of exclusion is applied in case of successful diagnosis of sarcoidosis. But there is no direct clinical marker which can diagnose the sarcoidosis condition. So just we have to perform a set of clinical investigations to rule out the active tuberculosis. Next. So this is the complete procedure to diagnose the sarcoidosis patient. Next. Another procedure is also, but that is also very risky. That the patients are given immunosuppressant. If the patient is responding to that immunosuppressant, that means patient is having sarcoidosis, because that is autoimmune condition and patient will respond. But clinician has to decide very carefully, because giving immunosuppressant to a TB patient is very
पावर बैंक मिलेगा भी
so disease will further progress. So a TB patient should not be given immunosuppressant. Our immune system should work. So that is why the diagnosis is very crucial. So one should be very careful while diagnosis sarcoidosis and TB. Is differentiating diagnosis and TB. Next. So that was an interest for us to see whether the serum metabolomics analysis, serum metabolic signatures can help us to differentiate the active tuberculosis from sarcoidosis. Next. Next. So when we compare the metabolic profiles using multivariate data analysis, we found that indeed the metabolic profiles of TB patients are drastically different compared to sarcoidosis patients, and the metabolic profiles of sarcoidosis patients are comparable with normal controls, whereas the metabolic profiles of TB patients are drastically different compared to normal controls. Next. So that was our discovery code. We also found there are many metabolic profiles which are significantly or which are distinctive to active tuberculosis and which are distinctive to sarcoidosis patients. Next. So these are the metabolic changes which we found in case of sarcoidosis and TB patients with respect to normal control group. Next. So among these various features, we also evaluated all for the diagnostic potential, means whether they can differentiate between active tuberculosis and sarcoidosis. And among all, we found that glutamate to glutamine ratio served very well, and it provided 100% specificity and sensitivity. Next. So we further in the second cohort that previously was discovery cohort, on the validation cohort again we found that this metabolic ratio, glutamate to glutamine ratio, consistently performed and provided us sufficient sensitivity and specificity for differentiating sarcoidosis from tuberculosis. Next. So this is another study reported in scientific report in 2020, where they have also shown the glutamate to glutamine ratio indeed has the ability to differentiate active tuberculosis from latent tuberculosis. So that also supports our finding that indeed this ratio has the potential to go for clinical settings. Next. So this is the result from their paper. They have also got 100% sensitivity and specificity for glutamate to glutamine ratio. Next. So coming to some biological introduction, why that happens? Why when there is active tuberculosis, so why glutamate to glutamine ratio elevates? So the reason behind it is that whenever the macrophage is infected by tuberculosis or pathogen, so that macrophage starts that glutamine also gets activated, that starts using glutamine at a very high rate, and inside that macrophage, the bacilli, in bacilli also this glutamine pathway